Hello everyone, this is your Biomedic Guruji with one more video. In this video, we are going to discuss the most important topic in biotechnology, that is regarding transformants and non-transformants. If they are transformants, are they recombinant or non-recombinant? To understand this, we need to first understand few things about E. coli bacteria. It has its own DNA which we call it as nucleoid, and it also has an extra chromosomal DNA which we call it as plasmid. Now, the very important point to remember here is the naturally occurring E. coli bacterial cells, they do not carry any kind of antibiotic resistance genes against these antibiotics such as ampicillin, chloramphenicol, tetracycline and kanamycin. These are antibiotics. Against these antibiotics, there are no naturally occurring antibiotic resistance genes. Okay. Now, if we were to insert the foreign DNA and identify it whether it is recombinant or non-recombinant, there are two methods. Now, in method 1, we are going to add selectable marker in the form of antibiotic resistance genes. So there are two types of selectable markers that we add here. One is ampicillin resistance gene and the other is tetracycline resistance gene. Now, in one of the cloning site of tetracycline resistance gene, we are going to add a foreign piece of DNA, which we also call it as alien piece of DNA. Then after the insertion of foreign DNA into the plasmid, we usually refer it as rDNA, and this rDNA is inserted back into the bacterial cell. Now, we don't know whether the bacterial cell has been successfully transferred, the rDNA is successfully transferred or not. We don't know that. So to find out whether they have transformed or they have not, we will be adding all the bacteria in the ampicillin medium. So it's a very important point, which medium is responsible or which medium shows whether the bacteria are transformants or non-transformants is ampicillin medium. Now, there's a Petri plate in which we are adding ampicillin medium and we will be adding all the bacteria. There's three possibilities. Now, to understand that, there will be a bacteria which is having no antibiotic resistance gene, then that means this is not at all transformed, and therefore when we add it in ampicillin medium, this bacteria is not going to survive and it will get eliminated. Such type of bacterial cell are referred as non-transformants. When they are placed in ampicillin medium, they get eliminated. The other two bacteria which survive is one bacteria with the foreign DNA and the other bacteria which is having both antibiotic resistance genes. By observing the transformants, we cannot 100% say that all the bacteria have rDNA. So to find out whether the bacteria are having rDNA or not, again we have to place it on tetracycline medium. So the second step is addition of the remaining transformant bacteria into tetracycline medium. Now, you should remember that if we added the bacteria with rDNA, the plasmid which is having rDNA, these bacteria cannot survive in tetracycline medium as the foreign DNA, as the insertion of foreign DNA has led to the inactivation of a tetracycline resistance property. So therefore, the recombinant bacteria, when added into tetracycline medium, they get eliminated. So the bacteria with non-recombinant condition can survive in a tetracycline medium. Then you might ask one question, then how can we select these as they are dying if we add it in tetracycline medium? The answer is here. So when we take ampicillin medium, we are not taking all the bacteria. So remember, this is a colony of bacteria. When we take the bacterial colony, patch of bacterial colony into the tetracycline medium, we are not completely taking the whole colony. We'll just take a part of the colony. So when we take this colony part and when it doesn't survive, we can say that this complete colony is having rDNA. So that is how we will identify rDNA, and we will take this patch of rDNA and grow it into the separate medium. Okay, so remember to give the conclusion part here. Non-transformants cannot survive in ampicillin or tetracycline medium. Transformants but recombinants will survive only in ampicillin medium, but they cannot survive in tetracycline medium. Transformants with non-recombinants can survive both in ampicillin and tetracycline medium, as it is having both the resistant properties. The second method is called as screening method. Here, the LAC-Z is going to act as selectable marker. The concept still remains the same. We are going to add LAC-Z as a selectable marker. That's a different selectable marker which we have selected. Now here only in LAC-Z only, we are going to insert the foreign DNA. So now when we insert the foreign DNA, the LAC-Z will get inactivated because of insertion of foreign DNA in its place. So that is what we call insertional inactivation. It's a very important point here. It has been asked in the NEET. So you have to remember, when a foreign piece of DNA is inserted in lac Z gene, lac Z gene will get inactivated due to insertion of foreign DNA, and that is what we call insertional inactivation. Now, all the bacteria after the insertion of foreign DNA is plated on a chromogenic substrate. Chromo means color, genic means producing. So color producing substance, on color producing substance, we are going to add all the bacteria. In the Petri plate, we are going to add the chromogenic substrate, and then we are also going to add the bacteria to see which bacteria have transformed and which bacteria have not, and which bacteria are having the recombinant bacteria or recombinant plasmid, and which bacteria do not have a recombinant plasmid. To see that, the bacteria, when placed onto the chromogenic substrate, they will produce color. So the bacteria which do not produce any color or show white color, they are usually referred as recombinant bacteria. Then the bacterial colony which is showing a blue color, they are usually referred as a non-recombinant. So the second method is very easy when compared to the first method, because in second method, just based on color, we are going to identify which is recombinant and which is non-recombinant. So this is how we are going to identify.